Hi, my name's Will Krawczyk, Scientific Services Manager at Chemical Computing Group. Today I'd like to show you how to make virtual mutations, and we'll do this using two different approaches. The first, using Protein Builder, which is a good application for making a single point mutation. The second is using Protein Design, which is a good application for making multiple mutations and analyzing the impact of those mutations upon affinity. So let's get started. The structure we'll be using today is interleukin 13. It's bound to an antibody. We see the antibody on the left-hand side of the main Mo window, shown predominantly with the beta sheets. Antigen sits on the right-hand side, predominantly with the helices. In the interest of time, I've already prepared our complex ready for interactive analysis, and I did that using Quick Prep. I've also annotated our antibody, both in terms of its atoms and the sequence simultaneously. So the first step of virtual mutagenesis is to decide which sites we want to mutate. Mo has tools that help us rationalize which sites are good candidates for mutations. As an example, I can use surfaces to calculate our electrostatics. I can also analyze protein contacts, and I can generate and analyze protein patches, and also predict protein properties so that I can look at downstream developability issues. All of these topics are covered in a separate video, so I won't look at these today. In this particular example, what we'll do is select methionine. It sits in a CDR. It's a good example of a residue that could oxidize over time if exposed to solvent. I'll just hit Site View so that we can isolate that residue together with anything within 4.5 angstroms. And now that we've got the system set up as we'd like, we can think about performing our virtual mutagenesis. Today we'll be exploring two different methods. The first is using Protein Builder, a good application for interactive analysis on single point mutations. The second uses Protein Design, a good application to perform multiple mutations and measure the impact of these mutations upon affinity and stability, whilst also calculating protein properties to assess any downstream developability issues. The first example leverages the Protein Builder application, so I'll just open that when we see it pinned to the left of the main Mo window. The panel comprises three main sections. At the top, a list of the standard amino acids, and if I click on more, you'll see the non-naturals, and by the way, we can customize these with our own non-natural structures. The middle part of the panel allows us to explore different conformations and minimize. The bottom allows us to explore different rotamers. Here I've already got the methionine selected in the main Mo window, and we see it also selected in the Protein Builder. Note the Protein Builder is set to mutate rather than prepend or append. So next we need to decide which residue we want to mutate this for. So here I don't want to negatively impact affinity or any other property, so I'm going to look for something of a similar size and shape, and for that reason glutamine comes to mind. So I'll just make that change, and we see that in real time in the main Mo window. However, this mutated residue now needs to be modelled in the local environment. So a standard workflow would be to first repack. So that's repacking the side chain and the environment fix to find the lowest rotamer energy. And then we can minimize. So I'm going to minimize the selected side chains only to minimize the mutation site with the side chain free to move and the environment restrained. An alternative option would be to minimize the selected residues with the entire residue free to move, which is a good option when we want to assess the backbone movement of a loop. So that's our first example complete. That's how we can perform a virtual mutation using the Protein Builder. Now in the next example, I'll want to perform the same mutation again on our wild type. So this is a good point for me to explain that there are unlimited undo functions in Mo. I could undo all the way back to the start of this session. Perform the undo to get back to our wild type methionine, so I'll close the Protein Builder, and then what we'll do is go ahead and explore the second example using Protein Design. You might recall that one of the benefits to Protein Design is that we can perform multiple mutations. So for that reason, in this example, I'm going to select not one, but two different sites for mutation. So I'll just select the neighbor. I'll go ahead and recalculate our site view, and then we can go ahead and launch our Protein Design application. So here we see the different operations that can be performed under the umbrella of protein design. Today we'll be using residue scan. I'll just click to open that panel, and we see here the same unified panel with these different operations that we also saw on the pull-down menu. So today we will be using residue scan. The panel itself allows at the top for us to specify our system and display settings. Then we get a mutation list together with options to customize these mutations, and at the bottom allows for scoring settings. So here I'll just select our first methionine, and you see that by default we'll be performing the virtual mutagenesis to all of the standard amino acids. But as I said, we can customize this, so I'll just clear that list. As an example, if I just want to make that single point mutation to a glutamine, I can do so just by typing it in. Equally, I can use the notepad icon to select from a list of the natural amino acids, also the non-naturals, or I could select them according to their property type. So as an example, I could mutate to all of the polar residues if I wish. So the second benefit to protein design is that we can assess the impact of these mutations upon affinity. So I'll just toggle on affinity measured relative to the antigen, which is fine. By default, we're going to be calculating our protein properties. So here you see protein properties, and the patches will be written to the output database. And with that, I'll go ahead and click build. So that opens a database viewer where the results will be written, but I just want to go back to the residue scan panel just to explain one thing, and that's related to this site limit. So site limit allows us to control how many sites are going to be mutated simultaneously. Site limit 1 means that only single point mutations will be generated, whereas site limit 2 allows us to generate both single and double mutants, and you see the mutation space grows accordingly here. So I see a database viewer has been opened. This is where the results will be written. The database viewer is the third major window type in Mo. It looks and feels much like an Excel spreadsheet, but if I just drag this field open, you see that it actually contains the 3D structure of our molecules. This is also our launching point to various other tools in Mo, so as an example, I can calculate some protein descriptors here should I wish. In this case, the database viewer contains the output from residue scan, so that application is repacking the side chains of the mutated residue and the environment. It minimizes the mutation site and it adds the model to the database together with scores. Notice that some of these lines here are mutating self to self, and the reason that we do that is so that we can establish a baseline to which the other mutations can be compared. So here you see that the values for affinity and stability are written to the output database, but I tend to look straight for the delta values. So delta affinity, delta stability, and these are measured relative to the wild type, which has a delta value of 0. So this allows me to very quickly identify those mutations which might worsen the affinity. So here we see a positive value, or improve the affinity. So that's given by a negative value here because it's an energy. Likewise, I can do the same looking at our stability values, and if I scroll along, you'll see that the protein properties have also been written to the output database. So here we have a descriptor. It's the size of the hydrophobic patches near the CDRs. It's an important parameter that's linked to developability, again measured relative to the wild type, which has a delta value of 0. So typically what I can do is then search and prioritize this output database according to the parameters which are most important to me. As an example, I could sort according to delta affinity should I wish. In this case though, I've just got a handful of results, so I'm just going to go straight ahead to browse through these so that I can examine these mutants in the 3D environment while simultaneously examining the quantitative data. So here I get a readout of the residue that I'm looking at together with the delta stability and affinity values. If I just move this to one side, I can also write any output parameters from the database viewer. So as an example, if I want to keep track of the hydrophobic patches near the CDRs, I can have that displayed in the browser. In the main Mo window itself, if I just hide our original co-crystal structure, I see the mutant shown here in green. I'll toggle on our wild type in grey, and from here I can assess each mutation virtually before I commit to any lab work. So I'll just click through a few of these. So here I see an example of a mutation that's generated a H bond. So we see that we're maintaining quite a good interaction there with our antigen. That's reflected in a negative delta affinity score, so that tells me that we've got an improved affinity, and it doesn't really impact the stability too negatively. So this is an example of a mutation that I might want to consider and moving that forwards through experimental lab work. And that brings us to the end of today's session today, where we've generated virtual mutations using two different methods. The first used the protein builder, a good application for performing interactive single-point mutations. The second leveraged protein design, which is a good application for looking at multiple mutations and also analyzing the impact of these mutations upon affinity, stability, and protein properties. Together, these approaches allow us to prioritize our experiments based on computational findings. I hope you've learned a lot today. If you have any questions, please do reach out to us at support@chemcomp.com.